So this is the first speaker for our Center for Biomedical Data Science. For those of you who don't know about our center, this was initiated by the Dean's Office directly earlier this year, Dean's Office of the School of Medicine. So in February, there was a Dean's workshop talking about why we need a Center for Biomedical Data Science and who we are, what we plan to do. And we also have a website. Feel free to go and take a look. Simply put, I don't mean to take over today's talk. I only have 2 minutes to give you the short announcement. So simply put, the center is here to serve as a hub for multidisciplinary talents across the campus, like you. We would like to facilitate the 3 Ps, in my opinion: people, program, and practice. So we would like to really bring the phenotype and the genotype, really have the translational medicine, really have to our way to facilitate precision medicine. We would cover training, education, research programs, and practice. If you have any suggestions, comments, and questions, feel free to contact us. Let me show you on this website where you can find us. So if you scroll down, first of all, go to medicine.eo.edu/cbds, and then you will see our mission statement, who we are, and what we want to do, what's going on at the center. If you scroll all the way down, you will see the event announcement and also Twitter. And feel free to follow us on the Twitter account at YieldData. So we'll announce things like this on Twitter. And all the way down, you have the contact us. If you click on this will show up on each page. So if you click on that, you will see the contact information. And then you can browse the member here. So right now, we have about 8 core members of the center. You'll see this, the leadership. It was originally directed by Professor Gersten and Professor Zhao. And I'm almost as new at the center. I joined about a month ago. So if you have any questions, feel free to shoot us an email. Right now we have nearly about 100 members in the center, and we're still growing. So this is, if you go all the way down, so this is just the first page of the center. Feel free to explore on your own. And before I end, I'd like to give some special thanks to Lisa Sobel. Without her, the seminar won't happen today, and she's going to provide wonderful snacks around 5 o'clock, so feel free to stay. So for those of you who actually are familiar with the CBB, which stands for Computational Biology and Bioinformatics Seminars, so we're going to change the name to CBDS, Center for Biomedical Data Science Seminar. We're going to keep it the same time. The first Wednesday of each month from 4 to 5, we're going to stay in the same room. So stay tuned. We're going to have a new speaker series coming up soon. All right, so without further ado, let Professor Smita introduce our speaker today, first speaker of the series.
Smita, for inviting me. It's really a pleasure to be here. In my group, we read Smita's papers and many of the papers of others in this room, so it's really fun to be here today and meet all of you. It's my first visit to Yale as well, so that's been really a great day. My talk today has a slightly presumptuous title. I apologize for that. Its purpose is, as a computational biologist, a lot of my career has been spent developing tools for analyzing data that biologists produce. But my view is that computational biology is actually a little bit deeper than that. In the sense that part of computational biology is not just fitting data, but actually sharpening the questions, to paraphrase Sam Carlin. And I thought I'd take the opportunity in preparing this talk to try to articulate a little bit about what we do in the single cell domain that's sort of more fundamental than just processing somebody's latest data. And that's my goal. I don't know if I'll achieve it, but that's the idea. And I thought I'd start with this slide. You'll see why it's relevant in a second. I'll tell you why I'm showing it. When I started my career, I started in mathematics and I became a math professor, and I was often speaking to mathematicians. And this kind of slide was how you'd start every talk. Tell the math people, all of biology. It's actually a figure from one of the famous textbooks in biology by James Watson, and like many of the things he says, it's actually wrong. Usually, this would be followed by let sigma be an alphabet of size 4, and then you're off to do math. The actual correct figure, the central dogma, is actually from before that by Francis Crick, where he explains that what he meant by, this actually simply he wrote out what he means by central dogma. I don't need to interpret it. It's the idea that information is unidirectional in biology, which is to say that there's no process that takes the information in proteins and copies it into DNA or into RNA. So it's really a statement about information. If you think about Watson's sort of characterization of this, which is incorrect, is the idea that mechanistically DNA makes RNA and makes protein, and that's just not true. Some RNA, for example, doesn't go on to make protein. And this distinction is sort of important because this statement is just simply a factually correct statement about biology. Watson's cartoon was simply an incorrect, very simplistic model of biology. But actually, already in 1957, people started to try to write down better models than what Watson came up with a few years later, and this is the first one I'm aware of. It's a paper from 1957 that wrote down a sort of very simple differential equation. Just to explain what's going on here, it's really simple. Here the idea is that you're modeling, actually I don't need to point it, but yeah, there we go. So you're modeling DNA being transcribed into RNA, and then RNA degrading, and so S here is the amount of RNA, and the rate of change is given by some production, which is due to the transcription, and some degradation rate gamma. This is a really simple thing. But just to give you an idea of how complicated things really are, in 2011, somebody wrote down a slightly more complicated model, still a very simple model, but just taking into account a lot of biology that was learned in the intervening 50 years. Namely that transcription is really just the first step in eukaryotes towards producing a messenger RNA molecule, or a mature RNA. There's a process of splicing, which involves removing these pink sequences called introns. There's capping of the molecule. There's a tail that's added, which consists of what's called a string of A's. It's called polyadenylation. And so all of these steps are important. And in 2011, somebody wrote down a slightly more complex model, which is that you have now abundances of unprocessed, therefore U molecules. They are being produced actually by the transcription. It's not really these end products, but the unprocessed transcripts. Some of them are being processed at a rate beta. Those produce the S molecules, which are the processed ones, which are these ones down here, and then those in turn are decaying. And okay, this is still a very simple, system of ordinary differential equations. You might ask, how can it be that the biologists know so little math that it took them 50 years to go from this model to that one? But that's actually not the right question. The reason that it takes 50 years to get that model is simply that there's no way to really collect the data that you really need to measure these parameters on a large scale for all genes until, basically 10 years ago. So, the model is simple because that's what you can imagine. It's no point to write down complicated models if you can't produce the data to actually study them. And I think the point is that we happen to be in a really exciting era now where we can actually start to measure all the parameters for all these models. By the way, you can now also imagine extending this model. Instead of just having degradation of RNA, you then have translation, not of all of the processed RNAs, only some of them. So there's a rate for producing protein from the processed transcripts and then some degradation of protein. So you can imagine extending this model. And I think we're in the beginning of an era where we're going to get much more complicated models that take into account a lot of other phenomena that are part of the system. That's we're really just in the beginning. So with that in mind, I think we can articulate sort of what we're actually trying to accomplish if we step back and think about this as computational biologists, at least in the sphere and domain of molecular biology. What we really have going on in biology is we have lots of cells. And the picture I just showed, that's what's happening in one cell. If you were actually paying attention, you'll see here that it says cytoplasm. This is actually a diagram of what's happening just in the nucleus before the RNAs get exported into the cytoplasm of the cell. So this is sort of, this is some dynamics that's happening inside one single cell. There's really a system like this in each and every cell, and those might be different. And so we're really looking at trying to learn the, model and learn the parameters for many, many different cells. These cells are not completely independent of each other. I've sort of drawn them clustered in this picture because these pair down here maybe are of the same type, and these up here are of the same type, and those in the corner are of the same type. But really, each of these cells is executing a complicated dynamical system. And really the question we're trying to get into, to go even one level further, is we're trying to understand how all these cells arrived at their states in which we're measuring them at some point in time from some precursor cells that possibly differentiated into these cells, maybe moving through some other cell states that we happen to have also measured or not. And so we're trying to solve for all of these systems and also their temporal order, making, as you can imagine, for a fairly complicated endeavor. In fact, just to give you a sense of what the scale is here, the number of distinct RNA molecules that exist, say, in a human is, we don't know exactly the number of distinct molecules that can be made in a cell, but it's sort of in the order of like 1 to 200,000. It's a large number. The number of distinct proteins that come out of that is maybe around 20,000, give or take 1,000. In each and every cell, there are at any given time about, we don't know exactly, and it probably differs by cell, but maybe around 300,000. And then we're looking at modeling a very large number of cells. The brain is of interest. In the brain, we have about 100 billion cells. So, and then we don't even know how many types they fall into. That's a question we're interested in right now. So we're a long ways away from figuring out, really having a model for all of this that's predictive and useful. But we're, for the first time, I think, in the position to collect the data for it. And what I'd like to do today is sort of some hints of that, with a mind towards thinking, a view towards thinking about the bigger question. So why are we getting there for the first time? So there's a field called single-cell genomics, and it's been around for decades. But it has traditionally involved various experimental techniques for isolating single cells and studying something about them. What's new right now is that in the last, say, about 3 to 4 years, there's been a series of technologies developed based on different ideas. Many of them are based on microfluidics, but there's some other ones that have allowed us to scale the probing of single cells. And in particular, this plot, which was made by my postdoc, Valentine Svensson, in his previous lab when he was a PhD student, shows on the x-axis your time, on the y-axis how many cells you can study in a single experiment with one of these technologies. And by study, I mean measure the abundance of all the transcripts, all the RNA. And these experiments are therefore called single-cell RNA-seq. And you'll see that, I think we're already in October 2018, okay, but just 2 or 3 years ago, we were seeing papers published where in one paper you had 100 cells in an experiment. And so what comes out of an experiment like this is literally, you should think about it, it's a matrix. It's got 20,000 or so rows for all of the proteins that can come out. Because you might not measure in the first experiments the RNAs at the granularity of individual transcripts, so they're agglomerated into genes, but maybe it's even 200,000 rows, depending on the granularity you're looking at, and then 100 columns, okay? And you measure a snapshot in time. To learn all these parameters of these dynamical systems, you're going to have to measure many, many such snapshots and for many more cells. Well, 3 years later, we're at 10,000 to 100,000 per experiment, and there's a data set now that's circulating, you can download it for free, that is made by a company called 10x Genomics that has 1 million cells for you to download. So you can imagine if you were a graduate student who started here at Yale in 2015, there's been just like a completely phased transition in the kind of data that can be obtained for studying these kinds of questions. And I want to say that what's driven this technological revolution in single-cell genomics and all of these technologies for measuring single cells is the drop in sequencing. This is a chart that's shown a lot like the first one that I showed. There was, right in 2007, there was sort of this phase transition in sequencing costs that made sequencing cheap enough that you could go and do these experiments. I'd like to talk about that for 1 minute. Specifically, I want to talk about this transition and what really made all of this possible. So in 2007, a colleague of mine at Caltech now, Barbara Wald, had the insight that DNA sequencing, although it was sort of primarily aimed at sequencing genomes initially, my genome, your genome, and so on, could actually be adapted to doing molecular biology by turning the sequencing into a means of counting in very high throughput. And she gave this analogy in a paper she wrote to Thomas Edison, who developed the phonograph as an answering machine. I actually didn't know this until reading the paper. And that was his intent, but actually it turned out to have a much different application, namely to play records. And so sometimes that happens with technology. You think that the goal is this, but it ends up being that. And that happened here. And her insight, I think, was really quite profound, because when sequencing is very, very cheap, you can measure various physical aspects of the molecular biology of cells by simply counting in very high throughput with sequencing. And these assays are called sequence census assays. Now I like to use the wildcard star, like in star-seq. She developed 2 assays, ChIP-seq, it's called, and RNA-seq in 2006 and 7. Nowadays, I don't think, not every people outside of biology don't think about this too much, but the vast majority of sequencing is actually not of genomes today, but is for these kinds of experiments, specifically RNA-seq, and measuring trisomy 21 non-invasively by sequencing pregnant mothers' plasma. So the way that these assays work, I think, is really kind of interesting and foundational experimentally, because these experiments sort of have a reasoning to them that is very computational. There's a sequence of reactions that you use to encode information in molecules. That's sort of the input part. And I'll show you in a second, I think it's very much akin to the way that in computer science you reduce one problem to another. Like, let's say when you're proving problems are incomplete. And so there's sort of a number of operations you can do. There's ways of modifying groups on molecules. There's ways of introducing site-specific mutations in molecules. There's ways of performing selective ligations, which means that you glue together sequences in a way that depends on the sequence itself. There's specific ways to cleave, based on specific sequences. And there's various techniques and strategies for pulling out molecules of certain types. So in a good lab today that develops this kind of technology, you need not really a good molecular biologist, but a good biochemist. A person who knows all of the chemical tricks and has a huge toolbox with which to combine these various operations. And really the imagination is the limit. There are now hundreds of different assays that measure everything from RNA abundance all the way to like an actual geometry of the DNA in a cell by using these primitives in clever ways. And you can imagine developing new primitives, and you can be creative and putting them together in new ways each time you get a novel assay. On the flip side, you have sort of a very simple way to decode the information that you put into the molecules. You can count events basically by barcoding. What I mean by barcode here is that you insert specific sequences in, which you then, by reading them out, you can count the occurrence of certain molecules. Similarly, you can insert sequences in the encoding step in order to distinguish objects. By measuring the actual sequences, you can learn about where the molecules that you're sequencing came from in some original genome reference. And you can find out where you introduced synthetically the mutations. So all of these techniques, and there are other things you can do as well from just sequencing, allow you to decode the information you encode. This is a really powerful concept. I sort of made this cartoon a few years ago to illustrate this. So you want to measure something about the cell, you become creative and reduce it to sequencing with all this encoding. Then you do the sequencing, which has become really cheap because there's been a race to sequence everybody's genome. So that's great. And then, what you get out are now these counts of these sequences that you've decoded. You need to go back and figure out the actual measurement that you encoded. And so usually there's some, statistics and computer science to do on that end. And then you analyze the data. This really has become now a really large enterprise within molecular biology. And all of these single cell techniques are really based on this idea. I just want to, for those of you who are not so familiar with this, quickly walk you through the basic assay that the Wald Lab developed. It was developed by Ali Mortazavi. The idea here is you have these RNA molecules, and you want to measure how much of them there is in a cell. Now, initially, she did this not cell by cell, but in bulk. So you're not going to be able to measure individual cells, but for a whole large collection of cells, you'll measure in aggregate, the relative abundances. So you want to estimate this pie chart. Again, you don't have just 3 objects, you have hundreds of thousands, but conceptually you're trying to estimate this pie chart. And for this assay, the biochemistry is not that complicated. You cannot sequence RNA, so what you do is you reverse transcribe the RNA back into DNA. So in fact, that's one of the main reasons that the Watson central dogma understanding was incorrect. DNA doesn't just go into RNA, RNA can actually go into DNA. So you can convert it into DNA. Once it's in DNA and you sequence it, then you can figure out which RNA molecules it came from because the DNA is a copy of the RNA. So that's the idea. It's a fairly straightforward assay. Just to be very clear about it, you have the soup which has these molecules, all these red things. You want to measure this pie chart for them. The way you do it is you pull, you make DNA, and then you pull out fragments out of this DNA. And I'm just going into this level of detail because, conceptually this is simple, but actually doing it is not that simple. There's a lot of biases in the experiment. You can't just read through these red molecules that you've turned into DNA. You can only measure parts of them. Your fragments have some length distribution that you need to be aware of that causes biases. You cannot read through even short fragments. You can read through a short fragment, not know what's in the middle, and then read a partner piece. These are called paired-end reads. That's the actual data you collect, and from all of that, you want to go and compare it back to the sequences you started with to figure out what you measured, and then you need to infer this pie chart. And it's complicated because some of these blue sequences actually could have originated from multiple red sequences, and you don't really know which they came from. So there's some math to do. That's called RNA-seq. The math itself is kind of interesting. Computer science, it's, as Smith was saying, it's a kind of project that my lab has worked on a lot. I just thought I'd mention it here because it's actually kind of interesting, I think, mathematically. There's a sort of fundamental object you build in order to do this. You take all your sequences, red, blue, and green here, and you build a graph from them that looks like this. It's called the de Bruijn graph. And for me, this was fun to return to this because I used to do combinatorics. In this graph, every node here is a short substring of these sequences here. And the point here is, the way I've drawn this, it's not so obvious, but this green piece here connects up with this green piece here. These are sort of the exons, if you remember from the second slide. This blue piece here connects there. And so each of these long strings is a path in this short, in this graph representation. One of the challenges in this whole area and domain is that the data sizes, they're not large by Google standards, but they're large by laptop standards. So you require some engineering. A typical experiment of this kind generates 20 to 30 million reads. There's, as I said, a few 100,000 transcripts, so this graph is large. There needs to be some engineering skill into going into how to construct these objects. Now what you can then do is, for each k-mer, or short substring in these sequences, you build this graph and you record simply which paths pass through that one. And then when you get your reads that you sequence, you can then basically hash them to this object and figure out where your reads could have come from. So it's simple but tedious to implement. We did this though, and one nice thing is that we could do this very quickly. And I mention that because I want to point out that we're moving very quickly along with the technologies in basically having this whole enterprise scale up to millions and millions of cells in an experiment. So as I said, given a read that we sequenced, we want to know where it came from so we can quickly hash it. And some of its k-mers are actually ambiguous to 3, some are ambiguous to a pair. It's sort of trivial to see that if you then just intersect these sets, then you can find the minimal set of paths that can explain your read. And by doing that, you can very quickly produce these counts, which are telling you for every set of transcripts that a read was ambiguous to how many of that ambiguity set you obtained. We call these transcript compatibility counts. And I want you to just remember that that's what comes out of one of these experiments. So what the basic raw data consists of sets of these transcripts. You're interested in quantifying them. You can't really do that directly because T1, for example, might appear in multiple sets. You have a multiplicity for each set. So there's some statistics you have to do in order to then infer the abundance. One way to do that is to maximize some likelihood function that you write down and use something like the expectation maximization algorithm. So there are other ways to do this. But basically, this is the raw data from which you then estimate a pie chart. Okay, so going back to this figure, I want to explain sort of its relevance to these assays. So remember I said you encode information in molecules, you sequence those molecules, you decode the information. So the way people have gone about doing 1 million cells and obtaining a matrix of abundances simultaneously for it's really like doing 1 million experiments. It's really like sort of mind-blowing if you think about it. Is to basically couple that encoding decoding idea to other technology ideas. And I'm going to give you just one example that comes from 2 papers in 2015 that use microfluidics. So let me just explain this briefly. So one of these papers, and I don't have time to go universally through everything, so I'm really just going to, of those 2 papers, pick just one. It's called DropSeq. And the idea here is actually pretty cool. So what you want to do, basically, is be able to sequence something at the end of the day that tells you that you saw some transcript and which cell it was from. And the way you do that is you're going to encapsulate your cells in a droplet, together with a bead, a synthetic bead, that has DNA on it, that is going to be a barcode for that cell. So what you do is you flow through cells, and you flow through beads in a microfluidic device. And I actually, to give you a sense of how big these devices are, I bought a little model I have. This is 8x times the size of the actual device, so the devices are tiny. But this is what it actually looks like, and these are the channels on it. You can probably see them easier here diagrammatically, but it's literally like microfluidics channels. These holes here are where you actually literally stick in a tube. Tiny, tiny tube, because it's 8 times smaller. But you stick in 3 tubes, and the tubes have beads, cells, and oil. And you're going to make, these droplets are going to be in an emulsion, like when you have oil and water mixed, you get little droplets. So what you do is you flow in the beads, things are coming from left to right, you flow in cells, and you set it up like, these are aqueous, and this green is oil, and so if you just set up all the timings just right, pumping in the beads and the cells in water, and then into the oil, you pinch off little droplets, and you have, it's just really cool, you have inside a little water droplet actually a molecular biology lab, because you're going to have a bead, a synthetic bead with DNA on it, and you're going to have a cell, and you're going to have some other stuff that will break apart the contents of the cell, and then the transcripts will get attached to the bead, and in this whole process happening, you're going to end up with sequences that have a transcript piece together with a synthetic barcode that says, oh, this came from this cell. And all the transcripts in that cell will get the same barcode. So when you do the sequencing, you're going to be able to decode and say, oh, these came from this cell, these came from that cell. So that's the idea. So this is sort of a blow-up of the microfluidic device that I just showed you. These are the channels down here. The reason that you have these tubes going up and down is to stabilize the flow. it's like doing plumbing, basically. And it turns out actually that there's a whole field of engineering around designing microfluidic devices like that and getting the emulsions to be monodispersed. It involves some interesting modeling. People have basically figured this out. How do you actually do this experiment? So this drop-seek is a sort of do-it-yourself thing. Usually you would buy the parts commercially, but one of the things we've been doing in my lab is actually building these ourselves. We've designed these syringe pumps, and I brought one with me. This is a $33 3D printed pump. And it's good enough and it's stable enough to produce these very controlled flows through this microfluidic device. It's relevant for me to this audience today for the purpose of my talk to mention this, because I want to sort of impart in you the idea that this is not some technology that's really complicated, that is very expensive today. This whole rig that we have, which contains a microscope so you can look at the actual flow and measure that it's all working correctly, and the pumps, it's for $300. So it's really, you can actually do it at home. In fact, I have one of these at home. And it's really fun. My kids love putting their clothing on and looking to the microscope and seeing the fibers. So it's really, really straightforward. By the way, for those of you who are interested in this, this sort of open source rig is the work of my student Sina Bursagi, who's a mechanical engineer. This started as sort of a hobby project, but he really spent a lot of time engineering this well, and we're hoping soon to publish this. It's all open source and free, and hopefully people will, some of you might find it useful. Okay, so the next thing I want to say is that, so it's basically these technologies have made this, this tractable very cheap, and I think that you should be sitting in this room, be thinking like, for those of you who are first year grad students here, some of you, you should be thinking, where will this plot be in 3 or 4 years when some other speaker like me comes, in October 2000, when will it be, 23 or something, who knows. They're going to show this plot, and there'll be a couple extra zeros here. So I think, what are you going to be able to learn, and what are you going to be able to model with this? One of the things before I get to the next slide, one of the things is that, when you have so many cells available for you in one experiment, then what you want to do is not just take one sample of cells and measure a lot of them. You want to measure lots of different cells in different conditions and under different perturbations, so you can learn parameters for these dynamical systems. And one of the things in my lab, so I have both, we work a lot on developing computational methods, but I have a wet lab now that we developed to do technology development. It's really a biotech lab more than a molecular biology lab. One of my students, now postdoc, Jay Scaring, is a really creative biochemist / molecular biologist, figured out a way to tag cells with DNA to add an extra layer of encoding so that you can pull together multiple samples. That's sort of a technology thing that allows you to take these large numbers and divide them into many, many samples to reduce the cost dramatically. So I don't have time to go too much into detail about how this works, but conceptually it's pretty simple. You have samples, you tag them with DNA molecules, so that in each cell now, in addition to the regular, the RNA that was there naturally, there's also DNA that we inserted synthetically, and that's unique to that sample. This allows us to then pool together cells from different samples. For example, it's really useful in cases where your samples have very few cells, where there wouldn't even be enough material to do an experiment at all. This kind of idea is actually not originated with us. It comes from any kind of cell, and I'm going to show you later like the kind of wide variety of cells we can actually do this on. This is Jace's magic biochemistry. The way we actually do this is in a one-step, one-click, sort of called click chemistry reaction. Our current protocol that we like uses methanol fixation, for those of you who care about that kind of thing. We're exploring other fixatives, and there's other ways to do this. But it's a really, really simple thing to do, and allows us to multiplex big experiments. Just to give you, a sense, before I get to showing you the results of doing some of this, it's really clean, this kind of multiplexing. and allows us to actually, we can multiplex thousands of samples. This is just a proof of principle we did where we multiplexed 4 samples and each of them with 2 tags. So just double 2 different DNA barcodes just to have some redundancy. And this picture shows the cells by the tags, and you can see that we have very clean data. Some cells have only this pair, and then they don't have the other pair. And you know, it looks easy on a plot like this, but in practice you have to make sure that you don't get leakage between cells and all sorts of other issues. So I really like this plot. This is called a TSNE, which just shows a sort of visualization of this data. I like it because sometimes in these microfluidics assays, you get unlucky and you capture 2 different cells in one droplet. Because it's actually a Poisson it's a random process flowing these cells through and the beads. Sometimes you get unlucky and you have 2 cells in one droplet, and 4 choose 2. You see our starting combinatorics, it's equal to 6. And so you can count 6 of the different types of doublets that you can get. Okay, so what can you do now with the multiplexing and the large number of cells? I should have said, you know, to do one single cell experiment today costs about $3,000. So if you want to do 100 experiments to start learning these models, you'd have to spend 300 grand. So the point of this multiplexing, and the reason it's sort of a big deal, even though it's a pretty simple technological thing, is that in my lab, I'm a math guy, we did 100 experiments, all right? Or 96, I should say. We took some neural stem cells in collaboration with the Thompson lab at Caltech, and we did 3 different types of perturbations. So we perturbed some factors, EGF, FGF, scriptase, and retinoic acid. So these are just ways to perturb these types of cells. And we did different extensive perturbations, sort of 4 levels. And so we produced these sort of 2 cubes of experiments. Each cell in this cube is an experimental condition. And some of them have very few cells. Some of them have a lot of cells. Some of them have few cells because you perturb the cells and they die. And now we're going to do for each of these conditions, produce a whole single cell matrix of genes by cells in that condition. And I'll show you the data. So you get a picture like this. So again, this TSNE is a sort of way to represent this high-dimensional data in 2D. If you're curious, I can explain it, but it doesn't matter so much for this talk. You should basically simply imagine that these clusters correspond to distinct entities in the high-dimensional space. So what's being done here is they've each been colored according to, excuse me, the different conditions that we were in. So you can see that the different conditions, I mean, sometimes they overlap and sometimes they put you in different parts of space. But what's sort of interesting is to go and now look at this by the types of conditions you're doing. So, I'm sorry, I said this incorrectly. Here, this is an unsupervised clustering, I should have said, where every color is just a distinct group of cells. This is the coloring we have colored by condition. And so what you can see is that in this condition over here, you know, you have you know, a lot of the cells down here, and you know, as you move, you know, this is you reduce one of these factors, you reduce the retinoic acid, you sort of shift over to this part of the cells. If you increase these factors, you shift somewhere up here. And so you can start to understand when you do perturbations how your cells are moving. We didn't fit any models here, but you can actually completely see how this is a very tractable thing to do. You know, this is just another view of this whole experiment. What's happening here is that, you know, we have these different clusters of cells. There are different cell types in these neural stem cells, and you know, we're looking at how much of each of these cell types we have in every one of the conditions we measured. That generates the relative abundances down here, and this is sort of a projection of those abundances of cell types. And so you can kind of, I don't know, it's a visualization we came up with. These cells right here in the middle are dead cells, basically. And then when you increase this factor, you move from this part of the space to that part. So you're changing the abundances of the representation of different cell types as you change these factors. You know, I think this is one of the largest experiments in terms of number of samples ever done, and really just the beginning. What I think is even more exciting is you can, you know, kind of take this idea now and I'll do all sorts of really cool experiments. And I think that, I give the title to the slide, A Single View of an Organism. This is an experiment we just did in my lab, in the jellyfish. And I think it's quite groundbreaking in the sense that with one experiment, we've sort of gotten a pretty far way into, in some sense, kind of starting to solve a whole organism. I'm going to show you some data right now that, like, just a few years ago would have taken the lab years to accumulate. We're working with the jellyfish. I never imagined in my wildest dreams that I would have a jellyfish in my lab. We have jellyfish. Why do we have jellyfish? Well, we have a collaborator upstairs, David Anderson at Caltech, who's interested to take this particular jellyfish, which displays certain behaviors, and to try to use it as a model organism for neuroscience. The reason he wants to do that is neuroscientists like to genetically engineer animals to fluoresce when certain parts of their brain are active. And they do this in mice all the time, but the problem is that a mouse is not translucent. You can't see into a mouse. So they stick needles into the mouse brain to then measure optically what's happening in the brain. Jellyfish is great because you can see all of the cells. Right? So the goal is, okay, challenge, take some animal that really hasn't been studied much. It's been studied a little bit by a group in France that sequenced its genome. Turn this into a model organism. What does that mean? We want to know, what are all the cell types in this jellyfish? What do they do? Which genes mark those cell types? How do these cells differentiate and develop into each other? All these questions. This would usually be a career for a biologist. We did one experiment, and I can't say we solved the organism, but we got very far. The specific experiment we did is we actually multiplexed 10 different individuals so we can study the variability in the biology of these different jellyfish. Multiplexed them, did a single cell experiment. This is, again, my postdoc, Jay Scaring-Smith, together with the postdoc, Brady Weisberg, and David Anderson lab. So let me show you sort of what comes out of this. This literally is an organism that nobody knew anything about. We get a big map like this. It's called Cladia hemispherica. We do some unsupervised clustering, which produces these colors. And now we go and ask, what are these clusters? What do they do? What do these cell types do? Now, just to give you an idea of the challenge here, in human, there are about 20,000 genes. And we know something about what they do because there has been a human genome project for 20 years or more. Part of it has constituted of having literally human beings functionally annotate each and every protein. What that means is they take a protein sequence. They compare it to all the known proteins. They look at the domains. They try to figure out what does it do. That's a huge amount of work. In this organism, there's some annotation that was produced by a computer, not human curated. There's some sequences. They make proteins. We don't exactly know if they're accurate. And we have no idea what most of these proteins do. You go and compare them. Some of them don't, some of them are unique to the jellyfish. But one nice thing is that if you want to study these cell types, we don't need to know for every protein in the jellyfish what it does. All we need is to find some genes that are uniquely expressed in one of these cell types and ask, what do those do? So we have narrowed down the goal of annotating the jellyfish, or this particular jellyfish, in actually a very targeted question where we just need to study a handful of these genes, and that's what we've been doing. So we've managed to annotate all of these cell types. We have the mouth, we have neuronal cell types, digestion cell types. There's a really interesting stinging cell type. And what we're about to do now is actually basically do what are called in situ hybridizations to see where these specific gene markers that we find for these cell types are actually expressed to basically align this figure with the actual cells in the actual organism. So one interesting thing that we can do is actually measure the dynamics a little bit, going back to the slide I showed in the beginning. And this is an idea that wasn't ours. It came in a paper in 2011. And the idea was that you can actually measure the parameters for this kind of a really simple system by doing gene expression measurements and comparing how much you have of the dark red or purple, I don't know what this is, dark pink or red, to the light pink. And see, when you have a cell and it's making these RNAs and you go and sequence, you're basically targeting to measure this, but you can actually either in a targeted way or by accident also measure the pink. And if you think about this model, the amount of pink is actually dictating to you how much red there will be in a couple of minutes. So what you're actually doing is measuring delta. You're measuring some kind of a derivative. And this idea is now called RNA velocity. It was recently published in a paper from the Linnerson and Korchenko groups. And this figure of it was made by my postdoc Valentin Svensson for this review, which recently wrote about this method. And basically what you have going on here is you have unprocessed transcripts and processed transcripts. And just conceptually, it's very simple. You're going to measure by sequencing some of these and some of those, and looking at the relative ratios, you can learn something about the direction in which your expression is going. So that's exactly what you can do here. And you can create a plot like this. This plot, I take with a grain of salt. It's not very quantitative. But this is the same picture you saw before of the cells. But you now have arrows sort of suggesting to you like where the cells are going in time. All right? So this is sort of a dynamic. It's sort of cool, actually, that you can take a static image and learn something about the dynamics from that. In theory, people have yet to really validate this carefully, but they did quite a bit of validation in their paper. This is our figure, and the reason I sort of became very excited and convinced is that you can see that for most of this figure, the arrows are very short, and right here the arrows are long, and these are actually the stinger cells, and there's a literature from this one French group that studies the jellyfish. The one thing they've done is look at these stinging cells, because they're constantly being renewed in the organism. They're constantly undergoing differentiation, so there's motion, even in a static single snapshot of the organism. You're measuring cells in a lot of different states as they differentiate to make specific genes that mark these processes, and it's actually kind of interesting. These arrows here, if you look at them, they point like they're slightly pointing in different directions. These are all pointing up, and these are going down, and we actually see two different genes that mark two different types of these stinger cells. Like you can see that we haven't solved this jellyfish by any means, but we're getting there quickly. We're learning a lot of molecular biology about this animal. We're currently in the process of focusing on the neural genes so that our collaborators can start doing their optogenetics experiments for studying behavior in this jellyfish. We're quite close to that. Just one word, I don't have a lot of time. One of the things we work on in my lab is methods for actually identifying, I just said, these genes mark this particular cell type. There's a lot of methodology that goes into that, and there's a very nice paper by my postdoc, Vasilis Dranos. He's an information theorist and my graduate student, Lin Yi. If you're interested, there's a paper in the bioarchive that we just put up recently that discusses that. I'm almost out of time, but I'll tell you one of my motivations in doing this jellyfish experiment was not just to help our collaborators, but I had a very specific question I was interested in. And one of the great things about having a lab is that we can now kind of do experiments to answer the crazy questions that I come up with in the middle of the night. So, so one of the questions I'm interested in is sort of a fundamental question about the variance of gene expression. So I showed you this picture before. This is the jellyfish and its constituent cell types identified by unsupervised clustering. This is the same picture actually colored by the individuals. Remember, we had 10 different jellyfish that we measured. And what you want to see is some mess like this, because I'm promising you that by pooling these jellyfish, we should have much batch effect. And so it actually looks really good. Each jellyfish represents all the different cell types. It's not like we have one cell type over here. It looks really nice. We did find one really cool thing, and we're still investigating this. There was one cell type, or I should say cluster 15, and it is represented much more in one single individual. So I don't know what's wrong with this particular, maybe this is the Harvard jellyfish, I don't know. But it's some weird jellyfish. Maybe, it's hard to imagine it's some batch issue. We had to develop the process of grinding up these jellyfish in order to be able to do this experiment, and it's basically you grind them through like a little, I know the word in Hebrew, what's it called, like where you pour your pasta through,
Strainer.
Strainer, thank you, Dan, yeah. So pour them through a strainer. So we don't think this is batch effect, but we have yet to prove that. what I wanted to show you here is you can kind of see that, this is looking at one gene that's, marking that specific cell cluster. We don't really, it's called XLOC. It's not that informative. We haven't really figured out yet what this cluster does or why it's in this individual. But we're about to go stain this gene and measure whether it's unique, one in 100, 1 in 1,000, or whether it was just an artifact. What I'm particularly excited about, though, is that for each individual and for each single gene, we can actually measure the variance of that gene in each individual and in every cell type. And so, I'm almost done. What I want to say is, so we can then ask the following question, right? For each gene, we can measure the average variance across individuals versus the variance of the average. Basically, the intrinsic versus the extrinsic noise. And I don't have results to show you about that yet. We've started to do this. We literally got this data only a few weeks ago. But it's sort of a very fundamental question, right? You're asking now, are there genes that are stable within a cell type, they're expressed, uniquely in a cell type and stable within there, but highly variable within the different individuals, versus genes that you see the same in every individual, but there's sort of a lot of variability by cell type. So, we are now in a position to basically study that. I think, I'd like to end 5 minutes ahead of time always for questions, so I think I'll end there. I'll just like to thank members of my group and point them out. So, Stina and Eduardo are my students who've worked on this pump project. And one of the great fun things we have going on in my lab now, Stina is a mechanical engineering graduate student. Eduardo is a biologist. They've been collaborating to do, not just to build this instrument, but also to do a whole other bunch of tech development for making single-cell assays better. Vasilis is an information theorist. Valentine got a PhD working on single-cell RNA-seq in Cambridge, but has a master's in mathematics. And Lynn is an MD PhD student who studied physics and computer science. So it's really fun. We have a lot of collaborators now that we're working with on all this. And I want to thank, these are my 3 co-authors on developing the quantification methods for RNA-seq that are implemented in Callisto. So thanks again, I'll stop there. Thank you.
I've got time for a couple questions.
Yeah.
One question. So when you tag the cells using the DNA, because we know that when we are processing those single cells, all the cells are under a decent amount of stress.
Yes.
And it may affect their morphology, affect their gene expression pattern. So I'm just wondering, if you add another process to tag all the cells, how much difference they would make the gene expression profile of those cells change? Yeah.
That's a really excellent question. What we've done is, I have a student working on this question right now. What we're doing actually is comparing, these cells are fixed cells, and they're also tagged. And we're comparing them to, standard single-cell RNA-seq where you don't fix the cells. Those cells also are undergoing various stress just to do the single cell. So it's not clear which clock is the more accurate one. But our first question was, are they the same? And to what extent is the biases in one or the other? A student who is working on this is Nadia Volovich. She's not done. I can't tell you the answer. I have looked at a bunch of data with her, and we have 2 different data sets that we're examining. One is we have it for the neural stem cells, but also another published data set. It looks like we're putting them through slightly different stress. We get fewer genes per cell, a little bit, measured. But it doesn't look too different. It looks pretty close. And I'm pretty confident that we can employ some techniques. There's a large, really a very developed literature of how to merge single-cell data sets. This is something Samantha works on, really good methods to do that. And so we've been using and testing those methods to see whether we can learn sort of a generic universal transformation for our data to kind of deal with this issue. And I think it's fixable. But yes, you're right. I mean, this is all to first order, and I'm confident that in the future we'll have better methods that don't involve shooting your poor cells through all of this stuff. And hopefully will also be more accurate. But I think not just our experiment, but there are many single-cell RNA-seq experiments now that suggest that, yeah, everything makes sense. We also have, for the neural stem cells, everything we saw was consistent with not only not tagging, but not also doing fixation. Yes?
My question is kind of twofold. What was your lower concentration for your samples, and then your upper concentration for all the pooled samples for cells?
Okay, that's a really good question. Yeah, I'll answer it like this. So, going into doing one of these standard single-cell RNA-seq experiments, you need, quite a few cells. What comes out of it is about 10, I'll quote, for example, 10x is the technology that does it. What comes out is about 10,000 cells, but you need some significant multiplier of that going into the experiment. The yield in 10x on cells is pretty high. It seems to hover around 50%. So if you put in 20,000 cells, you'll get 10,000. Drop-seq, for technical reasons that maybe can be overcome, but right now it has a much lower yield of about 5%. So it's cheaper to do this, and you can get the hardware for $300 instead of paying 10x, tens of thousands of dollars, but the yield is only 5%. When we do the pooling, the nice thing is that, so, if you wanted to run it, like let's say 10x experiment, just on a sample that didn't have many cells, you could do that. You don't have to start with 20,000. You could put in 5,000 cells and yield like 2,000. But there's some lower limit in a couple of hundred cells. Just because of just getting all the reagents working, it's not going to work. In the neural stem cell stuff, we managed to see samples where we only measured 30 cells. So there's no lower limit for us when we pool cells after tagging, which is a real advantage. I think, especially like you can imagine doing things like looking at embryos, which have very few cells, but looking at many of them. So some of them you won't get anything, but for many of them you'll actually get data. That's tagged by the individual. I hope that answers the question.
Just one question. So you showed a whole bunch of visualizations of different clusters of cells. So one of the challenges, of course, is how you define distances between cells to do clusters, and how you determine how many clusters or cell types are in there. I wonder if you have any insights or thought on what some of the challenges are there, how to approach that.
Okay, so first of all, I have a great, it's a kind of question I don't like to talk about, and I have a great way to punt it to say that some of the experts are here, and I just met them. Yuval Kruger's group has done a lot of work actually on this method, TSNE, both to make it fast, but also to study, whether there is some significance to how it's clustering the data. Having said that, I will answer your question. First of all, it's really important what the substrate is that you're clustering. Not every matrix is equal. Now, people right now, they're computing gene counts. That is to say, they take these reads and they align them to genes and they count how many reads did I get from this gene. But a gene is comprised of multiple different transcripts that also have different lengths. And I have a strong opinion that you should not work with raw gene counts, but with what we call these TCC counts, which are directly the counts of the sets. These counts down here, which are just, I saw this read ambiguous to these transcripts. I saw that 3 times. I saw this set ambiguous 5 times. And you can cluster these counts directly. And we've written a paper about that. When looking at this data, for various reasons, we like to work with the L1 one. I think that's like the right thing to do. But there are other things you could possibly do. Unfortunately, in this general field, people, not everyone, but many people just do something and publish it, and then that becomes a standard. So it's a little bit of a mess. And nobody, to my knowledge, has really carefully looked at to what extent it matters and doesn't matter and so forth, whether you use these counts or those, or, but it's a really, really important question. And one of the questions I was just talking about earlier today with somebody was, you might ask yourself an interesting question, which is when you have this kind of data, this plot is sort of a sin and illegal for reasons I won't get into. It's not okay to put these arrows on these dots. It doesn't make much sense. But it is sort of an interesting question, looking at this data, we're now in high-dimensional space. You don't just have a point, but you have some guess of where it would be in 5 minutes. Like, how should you present that? And what should be the right metric between this pair of points and this pair of points? How do you define distance between pairs of pairs in a biologically meaningful way that has some mathematical foundation to it. Yeah, so a really good question.
By the way, did you look at the data of this type for cycling cells in homogeneous population? How is it going to look?
For cycling cells?
Yeah.
Yeah, we've looked at that. You know, it's a little bit, so in the jellyfish we can't really do it because we don't really know which cells are like markers for, you know, the cell cycle yet. I mean, we have some guess, but yeah, in mouse and people have looked at this quite a lot. Yeah, and you can see that a little bit. You're asking me if we've looked at the RNA velocity, and that we haven't done directly, and I don't know if that's been done. That's a very good question.
The population is asymmetric.
Yeah, just looking at those genes.
Right?
Yeah.
So probably it will cancel each other.
Perhaps, yeah. I will tell you one interesting thing that we've looked at, which I didn't get to talk about, but we stumbled upon, is that when we do this tagging and we measure these tags, we can measure the number of them in a cell. And we sort of actually found that some cells get a lot of tags and some get fewer tags. And then we discovered that it seems to be proportional to the cell size. And we figured that out because we've done an experiment mixing human and mouse cells, and that makes a lot of sense. We are looking at this now because that's kind of cool. We have an orthogonal readout here of the cell size. And that should be also interesting to look at with respect to this velocity as well. So, yes?
Can you speak on what the single cell approach means for, say, single cell systems biology, like creating modules of the whole organism as far as its protein and its metabolome at the single cell level? Are there people doing that?
Yeah, so I mean, this is exactly what we're talking about doing here, right? You know, I think what it means to think about single cell from my point of view in systems way is that the basic unit isn't really the cell, it's the cell type. It's these clusters. And the value of the single cell data is to identify those. I don't think there's going to be much value in looking at every single individual cell. You know, we don't really care about cell number 1,345 or 1,729. You know, these cells are just really being used to identify the types. But then for each type, yeah, I think it's exactly what you want to do is you want to answer questions like you just asked about the systems biology. Now to do that, it's not going to be enough to measure the transcriptome. And there's already 2 papers out. One's called ReapSeq, and the other is called CiteSeq, and these are some of these seq assays that allow you to measure for a subset of proteins. The ReapSeq paper does 84, the protein abundance at the same time as the RNA abundance. So that's a next step towards doing real systems biology. And then you want to also measure the methylation state of the genome, and that, you know, nobody's done that. You know, when people ask me where's this field going, I think it's naive to think that it's all, you know, the number of cells, that's going to get very boring very quickly, but it's going to be in the number of assays you can do simultaneously in one cell, the multimodal way. And there's a lot of ideas of how to do that, and it's rapidly happening. Once that happens, yeah, I think we're going to get very far along really doing systems biology of the organism at the single cell level. We're going to figure out from one cell how you differentiate all the cell types, what they do, you know, yeah, all that's, and what would happen. Most exciting to me will be when you can say, hey, if I apply this perturbation, this is where we'll end up, because I have a predictive model.
All right, let's thank our Lior one more time.